Hello, Assalamu alaikum. This is the second video of the milk thistle is the silmarine. This is the anti-cancer mechanism of silmarine we will discuss. In the previous video, we was discussing about the anti-diabetic effect of silmarine. So let's understand about what is the cancer. This is the basically the cell cycle in which the G1 phase, which that require the CDK, cyclin dependent kinase and cyclin D and CDK4 and 6, and the synthesis phase S phase require the CDK2 and cyclin A, while the G2 phase require the cyclin CDK2 and cyclin E, while the M phase require the CDK1 and cyclin B. So in this way, the basically the for the cell division and the mitotic phase, S phase, G2 phase, and growth 2 phase and mitotic phase, and this is the G0 phase for the rest of the phase. But it will trigger if the this protein cyclin dependent kinase and cyclin basically when increase, so the more growth of cell and proliferate and divide more uncontrollability will occur. So the uncontrolled cell division will lead to cause cancer because repair will not possible. So the P21 protein 21 is a regulator specially will inhibit this CDK and cyclin B overexpression from the gene. So let's understand about the vascular endothelial growth factor is important for the angiogenesis, for the tumor progression, and the metastasis stage of the cancer will occur. This is the VEGF will bind with the endothelial cell, for example, for MAP mitogen activated protein kinase activate will lead to PI3 and AKT will activate lead to angiogenesis. So this milk thistle silmarine will inhibit this effect for the angiogenesis first. So the SLIT2 and the ROBO1 will trigger angiogenesis will also inhibit, while the BCL2 will inhibit the BAX. In this way, the apoptosis required the BCL2 will inhibit the apoptosis and cell survival, and in this way the caspase 3 and caspase 9 will trigger, but it will trigger through silmarine. So remember, and inhibit the BCL2 will not allow to cell survive and cell will die if the cell will inflame or damage. So also the reactive oxygen species will inhibit through silmarine whenever the inflammation will occur in the cell. So in this way, the reactive oxygen species will inhibit with silmarine, and the SOD, CAT and GSH will inhibit the reactive oxygen species also, and it will trigger also through silmarine, while the necrosis factor kappa B will trigger the TNF, tumor necrosis factor alpha interleukin 6 and 1 beta will lead to cause inflammation, and this will inhibit the reactive oxygen species through silmarine will lead to inhibit the inflammation, and the glycogen synthase kinase 3 beta will basically trigger via silmarine, and the beta catenin pathway will lead to inhibit the proliferation of the cell, while the mitogen activated protein kinase will trigger through silmarine will inhibit through silmarine will lead to inhibit the proliferation because it is the trigger. So the beta catenin will inhibit the proliferation, so it will be triggered through 9SK3 beta will trigger the beta catenin, and it is through silmarine, you can see in this diagram. And the PI3K will trigger the AKT to mTOR trigger, and this mTOR will inhibit the 9SK3 beta, but it will be triggered via a silmarine. I hope you make sense about that. See you in the next video. For this time, bye.